So here Gregor John Mendel 1822 to 1884, he conducted experiments on pea plants. He observed a number of generations and he understood the rules how the variations are inherited. So before we look into the work of Gregor John Mendel, first let us understand the basics of the traits, the variations or traits, the characters, how they are passed from generation to generation. See, we already discussed that every person, every organism is having two sets of chromosomes, two sets of genes that represent each character. So each trait is expressed by a gene that is a part of a DNA. See, in man, in his cell, he is having a pair of genes responsible for one character. Say for example length, that means height of the person. See, he is the father or the male. Height, he is having height. So he is having good height, that is some 6 feet height. I have written as a capital H, he is having good height. His height is determined by two genes, pair. The second one is, the second factor also determines the height, but it is small h, means short. But how one character is expressed out? Father is tall. Father is tall. Okay, better we take capital T for tall. So the tallness is controlled by two genes. The second gene is small t, means short. So a person is having two genes, one is telling you to grow tall, one is telling you to grow short. But only one is expressed out. He is tall. Means this is expressed out, it is there. He is having one more gene which tells that your height should be short, but it is recessive. The other gene is dominant, so that is why it is expressed out. For every character, a pair of genes are controlling. Even here, you see the mother, she is short. The height determining genes in her body, both are small t. Both genes are telling that short. So she is short here. So here, now there is reproduction by these two parents. So in sexual reproduction, they contribute their genes. Here the male contribute one set of genes in the germ cells, reproductive cells, not two sets of genes. So he has given this set of gene here. This set of genes. Father contributed this set of genes. It is small t. Okay, better we take this. Here the father contributed this set of genes, which is big T, and mother contributed this set of genes. Anyway, in both the cases, she is having small t. So here the boy, how he will be? Tall or short? Here he has got a set of genes. In that set, one gene is determining the height. Here what he acquired from the mother, in that set, the gene that determining the height is short. From the father, the set of genes that he acquired, the set of genes in that the gene determining the height is tall. So in this case, the boy is tall because this tall is a dominant character. Of course, the boy is having the gene which can make short also, but that gene is suppressed. The trait is suppressed by the tall because tall is a dominant trait. So here we can understand one concept, that every character, every trait is controlled by two genes in every organism. So if you understand this, we can understand the experiments conducted by Gregor John Mendel. That is the reason you might be wondered. So your father may be in fair complexion or dark complexion, your mother may be fair complexion. So you will get one. You may be very dark complexion you may get. That means you may acquire the character trait from your father. You may be very fair as like your mother. So you may acquire that fairness character trait from your mother. So the traits, they are expressed by a set of genes. So now let us see the experiments conducted by Gregor John Mendel. So now let us see the experiment that was conducted by Gregor John Mendel. Here we can see two generations, progeny 1, progeny 2, F1, F2 we can see here. So already I told you that why he has selected pea plants, because there are some characters which are visible to outside, which we can observe generation to generation, what changes happened, how the characters are inherited, we can observe it. So such characters are contrasting characters. What are those? Tallness and shortness. Pea plants, they have contrast characters. Some pea plants are tall, some are short. Some pea plants have round seeds, some have wrinkled seeds. Some pea plants have green seeds, some have yellow seeds. Some have white flowers, some have violet flowers. So these many variations are there in pea plants. That is the reason why he has selected the pea plants for his experiment. So here this experiment is based on the height of the pea plant, tall. So he selected a pea plant with tall and short, two pea plants. So these two pea plants are cross-pollinated. The tall pea plant, it is having capital T, means I told you, before this I told you that every character, every trait is expressed by two genes. One is dominant, one is recessive. So in this case, in the tall plant, both the genes are expressing tallness, TT. Both the genes are expressing the same character, tallness. Whereas the other plant, it has got two genes, pair of genes for the character, small t. So that is why it is short. So if cross-pollination takes place between this and this, the seeds that are obtained from these plants are planted. So in the first progeny, what is observed here? All the plants are tall, but for outside, just to look outside, all are tall. But let us see the ratio. Let us see their genes, whether both the genes are expressing tallness or not. No. One gene is expressing the tall trait, the other one is expressing the short. But the plant looks tall because the tall is the dominant character. The second case also capital T, small t. Third case also capital T, small t. Fourth case also capital T, small t. All the four plants, they have both the genes. The gene which is expressing tallness, which is expressing shortness. But outside only one thing is seen. A plant can never be short and tall at a time. It can either be short or tall. Here it is only tall because it is the dominant character. So now, here again F1 plant and F1 plant are crossbred for the second progeny. F1 into F1. How is that F1? Here this is the F1. In F1, two genes, one is capital T, the other one is small t. One is expressing tallness, the other one is expressing shortness. Here tallness and shortness. So now these are crossbred. So what is the result here? Out of this we got 1/4 short and 3/4 tall. 3/4 plants are tall. 1/4 is short. Out of 4, 1 is short. So the short plant has got small t. Both the characters are short. That is why it is short. In the other case, 50% out of 4, 2 are one capital T, one small t. Means one dominant character as well as it is carrying the recessive character also. Shortness expression gene is also there. Only one, it is having both the dominant characters TT. So in the second generation, 25% is having completely dominant, 25% is having completely recessive traits, and the 50% is having mixed, one dominant and one recessive gene. But to look outside, all the three are tall, one is short. So this is how the inheritance of variations takes place. So Mendel conducted some more experiments. In this case, he considered only one contrasting character, tall or short. In the next experiment, he has taken two different contrast characters. He considered tallness, shortness, at the same time their seeds. One plant with round seeds, one plant with wrinkle seeds. Likewise, in a combination of different contrast characters he has taken for the further experiment to understand better how these traits are inherited. Let us see. Now, let us see the rules of inheritance by taking two contrast characters. Earlier we have seen that only one contrast character, that is either short or tall plant, short plant. Here two. What is that? Here we are considering the color and shape of seeds of pea plant. So here in the initially, the seeds are taken like the seeds which are round. The roundness is determined by both the genes capital R, and green. It is determined by small y. On the other side, we have taken a seed which is wrinkled and yellow. Wrinkled yellow. The wrinkleness is shown by small r, yellow is shown by capital Y. The first one is round green. Both are crossbred. Here in the first generation F1 progeny, you got capital R, small r, means this is the dominant character. So you got it as round, capital Y, small y. The capital Y, it denotes that yellow. So round and yellow. Round and yellow seed you got in the F1 progeny, first generation. Now you have taken two same kind of first generation seeds and cross-pollinated, fertilized, crossbred. Crossbreeding has taken place between the F1 and F1. So in this case, the result is we get 315 round yellow seeds, 108 round green seeds, 101 wrinkle yellow seeds, 32 wrinkle green seeds. The ratio is 9:3:3:1. What does it shows? So the characters are independently inherited. It doesn't mean that the round and green, roundness and greenness both are not linked. So the characters, the traits are independently inherited. See here, the different combinations he got. Actually, he has taken round green, wrinkle yellow, but he got round yellow, round green, wrinkle yellow, wrinkle green. So this is the different combinations takes place because the traits are inherited individually. And the one more important thing is that each and every trait is represented by two genes.